G'day, good morning, good evening. I hope you're well where you sit or stand and listen. This is a medical cannabis program that talks about things related to the law. I respect the law, and I hope you respect the law too. And I only really promote organic growing practices. So that's the sort of stuff that I get into. Today's going to be cool. Where's the slide? This one. So today's on fungi and microbes, but more so it's on microbial research to see what's going on with microbes in relation to medical cannabis and the way that you consume. There's a really, really good study done on how pathogens can get through after smoking. After you light your joint or smoke it, it can actually maintain and withstand those temperatures, and it can follow through. There was so that's so important. So I'll go through that study a bit later, and you can see what travels through, and also for vaporizing too. Microbes can stay alive from the vaporizing process and enter into your lungs. So it's it was an eye-opening study for me too, because that's maybe a reason why I get some wheezing and stuff on my chest sometimes when I smoke a lot. It might be due to microbes coming through. So, all right, what other news is there? There was some amazing news for Australians. It's legal. They've legalised cannabis. G'day to Dave 1969, Levels Grow Room. How were you earlier? 96. Yes, hope you're doing well. Mate, it's so cool. I'm going to present. There's quite a few slides to get through to today. Entire screen. Momentic Meds, g'day mate, how's it going? I'm going to go on. I got some great news. Check this out. In Australia soon, there's a bill being placed in Parliament that's allowing half of Australia to grow 6 plants and show the cops 50 grams, 2 ounces. It's the same as ACT's law. So it's sweet. So I was that was only a few days ago. So it was, on the 20th of June. So apparently the Legalised Cannabis Party in Australia, they've gone and drafted a bill for Parliament to grow up to 6 plants per household in flower. So that's pretty cool. Oh, hang on. No, it doesn't say anything about flower. But anyway, it's 6 plants. It's terrific. So they've just gone on to say how much problems it is and how much 700,000 cannabis-related offences nationally since 2010, which really are possession offences. So, it's just ridiculous. Police, what do they estimate? It'll generates $8 billion in organised funds each year. Yes, all right. Anyway, so it's legalised soon, so it's really cool. So these are the 3 states. So it's Victoria, Western Australia, and New South Wales. Sorry about that up there in Queensland. And SANT, sorry about Tassie. Oh well, it'll come once they see it's happening in these states and all the positiveness coming out of it, and probably the money that they're more so interested in, the revenue that's going to generate for them. It's just bloody excellent. So he's a pro. He's for it. See, that was a story that was in the news made a couple of days ago. So it was excellent. Woohoo! Legalised Cannabis Party. Congratulations. Good work for Australia. Soon it'll be a lot more affordable, and I can start giving away some seeds, which will make a lot of people happy. All right, I'll stop the sharing there for a sec, see if anyone's rocked up. Oh, yep. G'day. Momentics, you were there before. I'll put on this back screen. Dave, tell me more about legal. Yes, it's going to happen soon, mate. Well, they're voting on it. So has it passed? Ask Cloudscape Mysterio. G'day, mate. It's getting voted on soon in Parliament. So after it passes, yay, I'll be doing a draw to make all yous happy. Keith Bonds. Hello, Keith Bonds. Good morning. Good evening to you. Supreme Great. G'day, mate. How's it going? Cloudscape Mysterio asks, means absolutely nothing unless it's passed. Yes, that's true, but it's a good start, mate. It means we're entering a grey area, which means like the same when cannabis was getting legalized in Canada when I was living over there, and enters a grey zone where if you're in trouble with it, it's going to be legalized soon, or not legalized, but it's going to be, well, it will be legalized, and they're asking for legalized in those states. So that means that once it goes to court at that time, it'll only be the most minimum charge, because by that time it'll be legal, and it could possibly be written in the law that all those charges are exempt, like they have in various countries. So that means that the grey area is so important, because cops basically just turn a blind eye to it because it's grey. It's going to be, by the time it'll get processed into court, it'll be virtually seen upon as legal now. So they can't charge you for anything legal, even though it was maybe they can pre-charge you, or they can just give you the minimum charge. Depends how it's written, how they change the law. But it's cool as. What's some other stuff? Good evening, Keith Bonds. All right, I'll show you all the stuff I've got to go through today. So it's a fair few. Fungi, this one. So there's all these slides to go through. So there's a few cool studies, and that one that study on inhalation of cannabis and what microbes are transported through. I'll go through that shortly. There's another ones on fungi, what comes out of your canopy, and the cleanliness in your grow. Is it beneficial to have leaf matter and things like that left on the floors and in pots and all that sort of stuff? So there's a rad in-depth study on that. Also on harvesting practice with fungi in relation. Should you do it wet or should you do it dry? Which one increases the amount of fungi growth? There's some other cool studies. Oh, just on the roots of how fungi can inoculate and improve the nutrient uptake of your cannabis plants. Anyway, that's a bit of an overrun. Oh, guttation. What guttation looks like in fungi. How cool is that? So yes, when the hydrothodes, when their pores have excess moisture that can't be volatilized off. Anyway, I'll get it back into it. We'll talk about it later. So this is a cool thing. Woohoo! Yes. Good on you, politicians in Australia. But this is all to do with Australian people too, with helping with all of their good activism and promotion. Not activism, you know what I mean. This is some sad news, but truth. I just put it in because I thought it's truth, but it's still sad. But we all saw that sub went down. But 5 very rich men went missing, and a multi-million-dollar rescue operation failed, and 750 migrants that are broke, and if they haven't got any, they're very poor, and they still have to tighten the borders. And it's just a contrast of interest, It's just anyway. Back to the positive. Here's a study. I was talking about them yet. There's more news to go through first. I know what the news was. There's news first before this stuff. So legalised. So the Health Minister in Australia refuses to fund the Australian Medical Cannabis Association's Compassionate Access Scheme. Yes. So his name is Mark Butler, the Honourable Mark Butler. He's rejected calls about the Cannabis National Scheme. So for those people who can't afford their medical cannabis, they were looking at it to try and put a compassionate scheme out. No, not going to happen. There he is there looking all, anyway. So that's about the guts of it, about that. So that was pretty sad for Australians. It's medical, they don't realise that we see it as it's so medicinally helpful for us in numerous ways. Everybody's got a different genetic makeup and put under different stress conditions, and it helps us in so many different ways. Oh, well, yes. And I thought I'd put this in because I'm into entrepreneurship. It's a way that I can work for myself because I have a permanent disability, so I can't work for anybody. it's too awkward and too hard, and I just can't. So I was very upset to read this. Australia's entrepreneurship. Economically, complexity ranked among the world's worst. Yeah, just the world's worst for our leading nation. It's supposed to be rad. It's politically driven too, because money is handed in different areas, and yeah, so it's just really sad, mate. Anyway, I'm not going to go on about that. There's a new cannabinoid that's with some major updates. So a few weeks ago, they found that CBC can regenerate the central nervous system of the human body. You can actually dead set regrow it. So this was a cool study done here that found those results. They called it, what's that? Cannabictrochium is the name for CBC. Like cannabidiol is CBD type thing. So this study, I'll just skim through it. What are neural stem cells? So it stimulates them. There might be a chart down here. Can CBC regenerate embryonic stem cells? A balancing act. It works in conjunction with THC. So THC downregulates the chlorine transmitter, which is involved in this system, while CBC boosts gene that codes for the special chlorine destroying enzyme. So the combination of the both of them, they work to improve, and they can regulate and protect the central nervous system, regenerate it. So it's cool as, mate. Cannabinoids, more better health for everybody. This is one of the studies. I suppose I might as well start because that was on a study before. This is a study that was it was published on the 13th of June 2023 in Canada, BC, and it was involved with Death Bubba. It was pretty rad, and it shows. Anyway, I'm just going to look at what it shows. Let's just talk about what happened. So there's to do the total yeast count, yeast and mold levels in these 5 cultivars that were tested. So they did numerous different ways on trying to test the different yeast and mold. Fungi is yeast and mold, if you break it down. Mold is flat. The mold grows with hyphae, with the little, the hyphae, they stick up, where the yeast is flat. And yeast is very colourful too, but it doesn't branch out and grow for hyphae, so that's how you can distinguish the difference to see what type of fungi family you've got. I should see plant materials. Here's the cool, this is how yeast can do it as well and do your own test. Actually, I'll see if there's anything said first, and then we'll get into that test. Devon Shipwreck. Hey, Devon Shipwreck, good evening. So we'll get back into sharing the total yeast and mold accounts and see how you can analyze yours as well. And you can also analyze it from color. There's really cool ways that you can see what's going down. So this study was done with flour. So they put it in a grinder. They grinded it for 30 seconds with water. They put it out, then they homogenized it. So they pasteurized it and get rid of all of the microbes in it. And then they can go and plate it. And then you can see the results here. And they've put it in, so you'll plate it and put it in for 5 days. And then after the growth, you'll check the growth and you'll see what's going on. This is also the test. Can I zoom in? Anyway, that's how to put it in your canopy. So if you're going to go and test yours, this is a way to test your canopy. So you get a clean petri dish and put it in there and just leave it there. They left theirs though for 24 hours. And these are the results. And from growing it, you can see the different colors. And what does the colors mean? Well, the yellow colonies, these are Aspergillus. The green colonies, they're Penicillium. The little brown colonies here, these brown colonies, they're Cladosporium. And the pink colonies, that's Fusarium. So then you can see what's just generalized what you know. And in cannabis, the main two they test for, when you see that they test them in the States and in Canada, they test for Aspergillus, the yellow, and Penicillium, the green. So that's what they mainly test for. Because then, so you won't know if you've got other ones. So if you've got other ones that are in there, they won't test for them because, there's thousands and thousands of different genuses of fungus. So what happened here? So these are the 3 different phenotypes: watermelon kush, pink kush, powdered donuts, Jack Herer, black cherry, and death baba. Woohoo! I work with death baba quite extensively, so it's good to see some tests done. And mine have a primed WRKY29 gene, which is a systemic acquired resisted gene I installed, and that's giving this low yeast and mold count levels. So mine don't, it's very hard, unless the immune system breaks down on it, it doesn't really show any. So that's pretty cool. So get down to the study. That's when they grew them out there, so you can see their efforts for growing. Yeah, it's just very basic. There's the death baba, so, and it's pink, so it looks a little bit like pink. And death baba doesn't look anything like it. It's too airy, the one they grew. Poor people. A little bit different conditions. Remember, you can grow it all different ways and bring it and express it differently. It's just whatever conditions you grow it under. Morphogelic markers. Now let's get down to some results. Compare morphogelic. No. Righto. I'd like to zoom back a bit if I could. Fresh tissue. So this is to do on the type of medium that was used. Potato dextrose, and there's some other different mediums that we used there to see which ones were they should proceed with. And you can see that's all equal, so it doesn't really matter. The substrate that you put in your dishes, that's simple. That's similar. Just seeing what amounts they got on it. This is pretty cool. This is electroscanning microscope of the different fungi, and you can see the hyphae that are stick up here. So this is the mold, and then the flat ones would be either the endospore or the spores that are still left there, or some yeast that they've zoomed in on. I think it's spores. I haven't read down there. Actually, look at this. So this is a test that you can do for your cultivars and see what you've got, see what endophytes you've got inside of your own. So if you get your stems, you'll cut them off, you'll dry them, make sure they're dry. So you've got your dry stems, and then you'll put them in favorable conditions for growing fungi and bacteria, fungi is above 70 degrees, 70% humidity. So just put it in a bag, put a little bit of water in there, not on this thing, and that'll just raise the humidity up. And as long as the bag's sweating, you'll see that the humidity quite suffice. Or to be more accurate, put a humidity meter in. Then you'll know it's working. That's what I do. And then you'll see what types of spores that you will come out, and they'll start to germinate. And you can see here where I've got the green one. So that means there's penicillium that were predominant endophytes in this cultivars. So you can do it and do the same thing for you. Here's a cool one. This is the study done with tests done to see how clean conditions are. Should you leave leaf matter on the ground or not? And should you shake the plant much when it's in flower? Well, those things promote fungal growth or spore growth. So here you can see down here, these two charts here. This one shows the no leaf litter. So this is in sort of sterile or good conditions. This is with leaf litter. So they put all this stuff on the ground and grew it. And this is with leaf litter and harvest activities. So you're moving the plant around, you're shaking it and that sort of stuff. And over here on the y-axis is the number of colonies that were grown in the petri dish. So you can see as they go up. So yeah, from you moving your plants, so there's rough people in harvest when they shake them and touch them, bang, they've just released spores. So, yeah, and also the cleanliness factor. Does it actually amount to spores being in the air and being airborne? Because they've got fans and stuff that are being grown, so that's how it's spread around. And the growing months, nothing to worry about. Actually, let's see if anyone said anything. Hey, Liney, cheers. I saw some CBC THC edibles in the shop last week and was wondering what their benefits were. Cool. Yes. Well, if you've got there, for those people with back problems or the neuron problems, what was it growing? Far out, I can't even remember. The neural tissue. Anyway. Yes. Gosh, I lost him now. So she's going to get some rad. Line up, huh? Oh, okay. All right, back to that cool as study. Total yeast. So this one shows the flower. Which is this one showing? This is a comparison of the total yeast and mold in the fresh buds that were grown under the same conditions over a 4-month period. So you can just see from already it depends on what cultivar, what genes are what's it called? What genes have resistance built into it, and what immune system, what was capable at the time. For instance, all of these might have had gene resistance put into it, but this one might have been very sick, and these two might have had a virus or something like that. But you can see from the tissues that it grows fine. So that wasn't the case. It was just a generalized study. So here's the total yeast and mold counts for the cultivars on the flowers, which is the black ones. So you can see that's where it was mostly contained. And on these little ones are the sugar leaves, and these smaller ones again are the fan leaves. So you can see it's not really much on the sugar leaves even, it's right in the bud. So that's because the moisture of the around the outsides is lower than the inside. So the water activity level required for the fungi to spread, to proliferate, is very higher. So that's the reason why that shows high. This is, it's not fair. Measurement of the climates. So it was pretty much the same climates were grown for all the same conditions, but they just wanted to show it just to have a thorough study. This is if it was a test done with fans and without fans, and you can see the massive difference, because you would have already guessed already. Without fans, it's not going to promote things, which without fans, the things things grow in stale conditions. So if you put a plant or anything under fans, you'll see the least amount of activity will be the area where fans are grown, where fans are located, and because they like stale conditions, because then they can just do what they want. That's like in these petri dishes, they just slowly grow and do their things because there's no air movement. So up here, the total yeast counts on the left, and on the x-axis is without fans and with fans. So with fans, it was lower than without fans. Good. Yes, that backs up my theory too. So, but everybody knows you should always keep the leaves a little bit nervous, just a little bit of shaking. So it also helps with, infections as well as their gaseous exchanging. And this is a cool test done to see what the results were on drying, on hanging. So should you do a wet trim or dry trim? What's the difference? And there's a massive difference. So from wet trim, look at them, they're just sitting there, no air movement. Dry trim, they're hanging. There's a possibility air's going around it and the humidity is getting evaporated. Here, look at this. Unless it's in a dehumidifying machine, fair dinkum. I'd like to see some humidity gauges in there. But anyway, that's what the results suggest too, that here we go. So you can see the blue is the wet trim and the orange is the hang dry. So it's really no comparison. The wet trim, it's just crazy. What's that? 5 to 25, 5 times as much yeast. So if those persons would like to harvest early and do wet trim, there's a 5 times effect that you're going to get, 5 times fold effect that you're going to have an increased microbial count in your. So when you go into cure or cure, it's yuck, it's already spoilt because your dry wasn't done properly. sad. Colony forming units, right? I think that's it. Oh, here we go. The processes of potential microbes contamination that can reduce post-harvest quality. So these are all the things that you can do to improve your post-harvest quality. So dead leaves on the ground, we've seen from the study, from the results above, that they release microbes back into the air, and anything can just travel around. Water also growing can harbour different and spread to different microbes, so that will become airborne intolerant and be spread through different ways throughout the plants. Applications of bioagents, so when you put stuff in, it will get on the plants too. Flower at harvest can be contaminated by tools and equipment. Mold in buildings can contaminate the product too, so it's not just in the building you're in, it's where's that air supply come from? Are there any cracks to draw drafts in? And endophytes, do you have endophytes inside of it, or do you not? Do you grow sterile? And do you have the right endophytes that are strong enough to counteract? Because remember, microbes have genes in them. They've got, bacteria has 5 to, I think, about 8,000 genes in it. So they've got to do different processes too. So if you've got the right types of endophytes that are inside, are helping you fight off stuff outside. That's it. There you go. What's this one? Scientists find cannabis compound inside a totally different plant compound. Okay, this isn't really related to fungus, but I suppose I'll talk about it. It's in news because it's only a week or so ago. This is in South America. They found that they found CBD increase. They found CBD is in this Trema bicrantha bloom plant. It's a shrub which grows across a lot of South America, and it's found to have CBD in the fruits and the flowers. So it's pretty cool. Doesn't say what amounts though, unfortunately. It's just a very brief thing, but scientists have previously found CBD in a related plant also in Thailand. So I've talked about it before, various plants that have cannabinoids in them. They're just in very low amounts, like in the points, like, what's that cool one? It's not one of the highest. Liverwort. Liverwort's one of the highest. THC. It's actually got THC-A in it. Right, I'll see if there's anything being written. Have a good one. No, nothing's been written. All right, back to it. Share screen. So I'll get into all these now. So I'll just start going through this stuff. So that was, this is a different study that was done that was seeing what the 5 major contributing factors were to reduce your microbial content. And it found that, what did he say, in Canada, did an amazing job in identifying the 5 critical variables which can impact fungal growth on the medical cannabis plants. And that's the number of bud leaves, air circulation, especially around when they're getting thicker from day 30, day 40 onwards, what type of harvest period you have related to a high temperature. Okay. Is it high or low? Yeah. Drying method. That was by the drying before you saw that the hang dry or wet dry. You can trim it fast, drying of moisture content, and the drying it down to what level? Do you dry it down to a level which can, remember, Saccharomyces cerevisiae can germinate at 65, at 0.65 water activity. So that's 12% in the bud. So if you were to spike it on the side, you'd see 12. You know that water activity is 65, and then that Saccharomyces possibly could germinate right in the center. So you've obtained a good level. That's today. This is the different types of fungi that there are. So you've got the non-colonisation. So this doesn't get in. So arbuscular mycorrhizae or endophytic one, which goes inside of it. This is what it does. And then if it's on the outside, ecto, it doesn't colonise it. It does this. Like the endophytes, they scavenge nutrients from saprophytes. Like saprophytes are things that decompose things, and they can get all the nutrients that way. I should show actually the difference between them. Geez, I'm confused now. I'll just read this one. So the endophytes, or the arbuscular mycorrhizae, they increase pathogen resistance. They help to a lot of resistances because they're inside of it, and they can activate the pathways that help with systemic acquired resistance. That'll do. That was that cool study before. That was such a thorough study that it was just really good to know what goes on and what they've done and how you can test your own. This is a nice little experiment grown with a plant inside of a glass box, and it was testing blue Athrospira platensis and Rhizophagus regularis. What was written about that? So a small pine tree grown in a glass box shows the level of white finely branching. Okay, geez, I'm confused. So this is just an example of its branching. Sorry, it must have been a different one. So you can see this is related to probably a drip pattern too. So this might have been dripped in, and that creates that wetting pattern around there because it's adventitious roots. It's not a really long taproot. But actually, we're talking about fungi today, so I'll try and stick on topic. These are some forest light mushrooms that are bioluminescent. How cool is that? So they've got that bioluminescent gene in it, and it activates. That's something I'd like to work with, that's for sure. This is the planet's geography of the fungi. So we can see what's beneath the soil and see what's going on there. So there's a map of the world there, and you can see here's on the latitude and the longitude. Sorry, the longitude's that way and latitude's around. So this is on the sides broken down. So you can see on the equator's in the middle, and the Arctic's regions are at the tops and the bottoms. So you can see for the ectos mycorrhizae, it's very prominent inside, but not at the equator. So I'd like it being inside the Arctic plant tissues, but not around the equator. And the arbuscular mycorrhizae is a bit different. And the nitrifying is, well, as you'd expect, it's around the equator because there's not much sun sometimes at the opposite ends. So this is the endophytes that are inside. So you can see that they like to hang around the equator region because this chart here shows brown is high, blue is low. So that's for all of these. And the ecto, the ones that all the fungi that are on the outsides of the plants, they are very high in the polar regions, so Antarctica, but no, that suggests it might be, but it doesn't show brown. All right, 2019 that was from. What's the role of fungi in the soil? I put this in because it was sort of talking about fungi. It's a mysterious, no, I'm not going to go and read all that. So fungi just really assists with putting out branching because it can extend its hyphae very fast. It can multiply fast, where plants' roots only multiply every 15 or 16 hours, say, for cannabis. So this can branch a lot faster. So it'll grab, depends what type you've got, to how it's going to symbiotically work with medical cannabis. But when they do, they can transport nutrients faster and make nutrient uptake faster for cannabis. Oh, this is a little, what the membership thing does. So $5, so support. So I appreciate those people that have done that. Thank you very much. It helps. $80 membership gets you about a dozen private laylists that I've got. All my playlists have thousands of lectures that I've got in that are related to plant, soil science and microbiology for free. You're welcome to go through. $150 gets a 20-minute video call or a novelty gift like a chart or something that I've made. And $300 is more so for a company or for a person going through hard doctor's programs and wants my educated opinions. This is that rad study. If today's topic, this should have been talked about at the start. It's a bit of a shame, but it wasn't. This is a rad study that opened my eyes to do with cannabis smoking and what microbes are around post-inhalation or post-lighting of it and stuff like that, like in your bong. They just harbour heaps because there's water in there. So the water activity level's high enough to keep growing stuff. So that darkness, it mightn't be black tar sitting there. That might be microbes or fungi spores growing inside of it if you put it under a microscope. So there's only 7 slides here, but it's so important about this, acute and chronic risks. So it just shows viable microbes and their metabolites are found in smoke, believe it or not. Both living microbes and non-living biological agents are associated with acute and chronic diseases found in cannabis consumers. Inhalation of bioaerosols possesses unique risks to smokers. Cannabis users acute infections and pathogens in smoke. So there's a test done here where they hooked up a Hepa filter, a blimmin' Hepa filter, to a cigarette, and they saw what was found in it. So they extracted the Hepa filter with some liquid, with 1 milliliters of liquid, and then they put it through a sequencing machine, and they found what was actually in there. And believe it or not, things survived past the Hepa filter. Viable microbes survive combustion and vaporization and carry over in tobacco and cannabis smoke. The studies here, they showed that the Pseudomonas, Cryptococcus, Rhizopus, and Aspergillus species were associated with cannabis that they found post-smoking, post-combustion and vaporizing. Mate, this is bad. This is not. So for those that might equates why sometimes I get a bit of wheezing on the chest, because maybe some more spores were around at the time of my consumption and that this process didn't get rid of it, which I thought it used to. Cannabis users are 3 to 5 times more likely to have fungal infections than non-cannabis users, and 43% of infections attribute to Aspergillosis, which is, Aspergillus, growing inside of you. The harmful biological agents produced by microbes. So inside of the microbes, they bioaccumulate things as well. And they have toxins that are in them. And when they're broken, they release their mycotoxins and can kill things, kill our spores. This is this process here. So pyropoptis is the cell's death. So it impacts on the immune system, reducing it, and then it can actually kill some endotoxins get in there and they can actually kill the cells. Microbial byproducts, they're endotoxins like exotoxins, ergosterol, peptidoglycan, and other components. And peptidoglycan, that's the cell walls, that's the constituent, that's what bacteria cell walls are made up of, is peptidoglycan. Like the cell walls in plants are hemicellulose, cellulose, and lignin, where in bacteria it's peptidoglycan. So that can have microbial products that are bad for us. So when we burn them or they go through our consumption process, that cell wall breaks and it can cause this process. This is its individual actual pathway in which it goes through. So I'm not going to go through all those different pathways, but that's what it ends up in, having asthma or CPD or lung cancer. So these are all the different studies down the bottom that have been done to get to these results. So the hundreds of mycotoxins that are found in fungi, there's a few of them here that are described. I'm not going to tell them, I'm going to go out in the big names. And some fungi and bacterial allergens. So pediatric allergy and immunology. Oh, okay. So that just goes through some different, I'm talking about that one too. Next one. So biological agents in smoke linked to chronic pulmonary diseases in cannabis users. So that shows how it's linked and the pathways it goes through. It goes right into the bronchioles and bronchi, right up real deeply up in there. Excuse me. Smoking cannabis is associated with more outpatient medical visits in respiratory issues. This is more so meaning that with fungal, you need to have fungal resistant genetics. That helps not germinate them and make them proliferate. That will help beneficial fungus protect the plant, I suppose, and not let these harmful ones that could possibly proliferate in. So ways that we could move forward would be to, actually, I'll talk about that at the end. More studies down there you can go and access to see where they got to these results. Process preventative controls in food safety. Okay. So not really going to talk about that. That's to do with food safety. So no kill steps validated for flour products. Okay. This was to talk about how when they process foods, there's all these sorts of measures that they put into, 5-step measures that they put in to maintain no microbes are coming out in food. Cannabis at the moment has 0. So this was just stating that how important it is for medical cannabis probably to change in the future to implement this method. Culinary and medical herbal product guidelines. What's this do? Fresh culinary herb food safety programs should focus on preventing adulteration by microbial contamination, because these herbs are often eaten raw and without kill steps. It's more to say that better handling practices, being more aware about the things that we can't see on the plant. People only are aware of things when they see it. They think, shit, look, it's dying, or there's spores in there, or there's color on it, or what's going on. But they're always there because they're in our air. If you breathe in now, they're always there. Smoking cannabis, this is a summary. Smoking cannabis has both living and non-living microbes associated risks with it, like acute and chronic pulmonary diseases, and all hazards should be considered. Cannabis microbial reduction technologies have not been scientifically validated. The critical limits have not yet been established to reduce viable pathogens, and they do not remove non-viable microbial agents. All they do is they've got a UVC machine that when you build a licensed producer, will have to have a CVD machine, a UVC machine in their budget. They've got to build it, put it on site, and if their product has agents in it, they're allowed to zap them. And as long as it comes beneath, I think 5%, don't quote me, but it's beneath a level that's acceptable. So they can start at 15% or 20. Not 20, that's too high. But they'd start up, high, and they can actually keep zapping it and reduce it and try and make it to acceptable levels. That's what's written in standard at the moment. The best approach is to cultivate with good practices to produce pathogen-free, low bioburden chemical cannabis products to protect consumers from acute risks and chronic diseases. So it's all those methods that we talked about from that study previously, about drying it, about handling it, about conditions that it's in. There's so many things that this sort of is making it very technical. But it is technical if you want medical cannabis. If you want cannabis or street marijuana, no worries, mate. Don't do any of this. But because I'm into strict medical cannabis, where it has to be, it's for a medicinal purpose. I want to improve my health. I want to treat symptoms and reduce pain and etc. So that's why I went to good medical practices. And to summarize too, this was written by Rogue Micro, ROGUE, MICRO was this talk given. So for me to have shared it, I had to have said that. So props on that, on safe consumptions for medical cannabis. I hope you can do it too. Can of jobs. What sort of can of jobs this week? Here we go. So what's all those folk? Reason this is put in is because You all smoke cannabis, and a lot of people don't have jobs because they're disabled or they have their cannabis for a reason, so they can't work. So this is entrepreneurship that I put in, so anybody can possibly get jobs even if they sit at home and do nothing. So let's see how you can work for yourself. So if you're artistic, you can get online and do artistic stuff. That's why this was put in. Cannabis catering. So for those who it's legal in your states, territories now, your regions, provinces, you can go and register cannabis catering businesses and try and do this type of stuff. Here's another cannabis catering. How cool is that? Just looks, look at that. That's just all that's just leaf, not even trim. And it looks nice because they've artistically made it look like that. There's no buds in that photo even. It's very nice, artistic, well done. Cannabis wedding planning services. I was amazed to see how cool that was. Just giving a twist on it for all those people that are right into it, like myself. Look at that, you can throw a big bouquet of buds. Then you have a nice cake afterwards with wow. I think you'd like that even if it's pretty fancy looking. Look at that, chunks on top. Wow, that's pretty big. There you go, there's a nice bouquet. Yes. So that's for the cannabis wedding planners. This is for the person who's into fine artwork, whether into metal or castings possibly. You know, you can go and get all of your things and make a silicon cast out of stuff. Just mix some stuff, get a silicon cast kit, mix them up and pour different metals or whatever you want into them and pop this out and then sell it. Some ideas for those who have nice stuff around. This is artwork. How rad is that? Look at that. I like that. Follow the yellow brick road. That looks pretty spiffy. Wow, those nice colours. Yes. Into the cannabis jungle. Looks nice, nice walkway. Exterior design, persons could probably get heat off that. How gut and bacteria affects brains and bodies. I'm actually not going to talk about that because that's not really related. I'll wait for a microbe one because we're talking about fungus today. This is bacteria. I'm going to talk about fungus, I think. So we'll skip that. Here you go. This is how fungi establish itself outside. Where does fungus grow? How does it get round? Well, it takes many generations for it to establish itself successfully because it gets down and it grows all the mycelium underground, like when you have big forests. If you lift some of that up, go down a foot, 30 centimeters, take a shovel up, you'll see a big carpet mat of white stuff. That's all the mycelium. So you can see the times for it to grow, it establishes really well in your old forests and your new areas or grasslands. It's bacterial dominated soil. So it's not really going to proliferate there. It needs all of the decomposition and breaking down and that sort of stuff. Like it gets over in here. And as time ages, that's what happens. Everything breaks down, drops on it, and it builds up and builds up. The O layer on your soil horizons goes up, and your A layer as well. And yeah, you get your nice fungi dominance. This is cubensis growing in coconut haws, reaching out their heads. I'll put this in to see the times of the fruits. So this is just ready to throw fruits out. So just under that soil before, you can see up here, I was talking about the mycelium growth. When does it breach the surface? Well, fungi is 4C to 14 Celsius, 40 Fahrenheit to, what did I say, 14? What's that, about 70, 65? 65 odd Fahrenheit, that's its conditions that it likes. That's for most cultivars. There's other reasons out of that. It does branch, it does flower in. And that's right when here. So it sits under the surface until it gets its right conditions, and then it starts putting its fruit out. So these are what it looks like underneath, just before it's ready to fruit, and that's where it's starting to fruit there. There's another little cubensis. I do support cubensis mushrooms. The sources of, yeah, that's right. We went through this before about the different fungal contaminations you can get from dirty or how you can get it in your cannabis plants. Oh, this is cool. I just like the Abos. They're cool. I support the Aboriginal people and all the native people around. They were here first, and I have great respects for them. And I just like this because it's just a good way to, and I just like how they drew Tasmania. Yeah. So to all those native folk out there, cheers to yous, cool people. I feel sorry for yous, how people have just gone and taken your lands and resold them. Anyway, I'm not into. This is cutation. So this is the red light sap that comes out of fungus of these mushrooms. And it's called cutation. And the cutation happens because the pores in here are usually releasing moisture, and they can't release the moisture because outside here is too humid. So it means that the conditions outside are, say, fog-like or cloud-like conditions, and this can't be evaporated. So it just slowly builds up in a bubble. So cannabis gets that as well. That's called cutation. It's a good way to know that your clones are ready, actually. You'll see if it's got cutation happening, it means there's a fair bit of rooting that's happened underneath. That's for clones. Was just not for this. This is just a cool looking thing. And see that, yeah, that'll do. Total yeast and mold counts. Actually, I went through that, didn't I? Yes. That was talking about the different colors, how you can do it. Yep. Oh, yeah, that was that study. Oh, this is really good. This is the water activity levels. So it gives you an idea of talking about water activity. I was saying before, you want to finish your cannabis when you dry it between, what was it, 12 and 14% is the maximum you should finish it at, which equates to 0.65 and 0.70 water activity, which means that that's the lowest amount that fungal spores can germinate. Saccharomyces cerevisiae is 0.65. So if you put your little probe into it, your little moisture meter, stick it in the side. I've made videos on how to do that. You'll see that if it's above 13 or 14, keep drying it. Otherwise, you'll have a chance in growing fungal spores, them germinating. This is a comparison of water activity in some common foods, so you can know what's allowed. So dried milk, they have to get down to 0.7 because that's the minimum that the fungi can grow. Now I'm going to have to bring that up. I wonder if there's time, actually. Is it in here? It's not. These are some, I'll just read it out. So cheese spread is very wet, 0.95. Soy sauce, which is wet, 0.8. And then you come down to dried milk, which is 0.7. Or peanut butter with a 15% total moisture equates to 0.7. So it's got to get down to lower than that to containing water activity. This is a water activity monitor. So it's just an example. In other words, dry milk, you think, wow, it's got no moisture in it. It's got 8% moisture in it and 0.7. So it just adds up. Oh, this is a nice looking, this is not photoshopped or anything. This is a wax cap mushroom, a parrot wax cap mushroom. And it's just a nice photo of it. I thought, how cool is that? It just looks really nice. It's like as if someone painted it, but they haven't. That's the end. That's the end. Oh, no, I haven't gone through. I'll see if there's any comments or anything. Raptor. How are you, Raptor? A bit late, mate. It's a big fungus special. You would have loved it. You should have been up here. Would have let you come up. Main Green Girl, how you going? Nice to see you too, my friend. And it's been 55 minutes. I can go through now I got a little section on fungi. my own personal one. We'll see what's in here. This one, because I've been through all my slides today. Yeah, let's go through it. Little quick lesson. This is old stuff, so this will be basic. Mycorrhizae, symbiosis, mutual effects. There's different types. Yeah, I went through the two different types, two different main types. There's other types, but I went through the two different main types. Vascular, yeah, went through them. Ecto, they're the exterior ones. We work exterior. Endos or arbusculars, the insides. That's the main difference. Why mycorrhiza? Well, it works, it extends the root hairs, basically. It's increased surface area. There you go. The fungi has the following features. It's spore-bearing, it's eukaryotic cells, it's not a prokaryotic, so it's the same human cells as us. It breathes the same as us too. They do not use CO2 as a source of carbon. It's interesting. That's why they respire CO2. They put that out. And for those cannabis growers thinking they have enough mushrooms in the area to satisfy their girls, you have to have twice the amount. If you want a 4x4 of your grow, you're going to have an 8x8 of cannabis respiring to give it a 1300 CO2 to 1500 CO2. Fungi. People can use fungi to produce food. Oh, really? Oh, wow. Okay, I shouldn't be smart, sorry. Fungi can cause organic matter to decompose. Yes, they're brilliant saprophytes. That's what that means. They put out their enzymes and slowly start breaking it. Oh, yeah, look, fungi are saprophytic, which means they obtain nutrients from dead or decaying organic matter because they put out their own enzymes, and you've seen it all jello and melting around it, and they slowly absorb them. Fungi store carbohydrates and glycogen. Most fungi are aerobic, though some yeasts can be facultative anaerobes. So that means the anaerobes has no oxygen, and facultative means it's not an obligate anaerobe. An obligate anaerobe means its obligation uses one thing. Facultative means it can do multiple things. So it has multiple sources. The mushrooms grow in the grass, on the trees. Okay, I'm not going to read that. Classifications. Oh, yeah, there's a few different classifications of their. There are 7 main phyla and 1 subphylum. Actually, I'm not going to break it into that much. That goes in. This is going hardcore into it. Goes down to the different families that they're in. The protistas. These are protistas. They're not even fungi. They're a mixture. Algae. Yeah. Sorry, I'll just get to. I'm sure there's more fungi stuff. Earth, water, and slime molds. But in my setters, I suppose I should talk about them, hey? Because they're all right. Because it said umycetes there, and the umycetes are the Fusarium and Pythium, and those types of bad ones that cause problems for cannabis. So I'll just I'll briefly touch on it. Not algae though. Next is some beneficial. That's cyanobacteria. The difference between algae and cyanobacteria is their cells. So algae has prokaryotic cells, and cyanobacteria has the single-celled, which is very beneficial, prokaryotic. I want to skim over this because it'll just bore people, I think. There's a diatom. That's where diatomaceous earth comes from. Green algae. Where's the pathogenic ones? They're waterborne. Protozoa. No. No. Amoebas. They're so unhealthy for us. They're in sludge. Actually, we're talking about the arrow branch of it. Sorry, I really get distracted a lot and like to go off and just keep describing things and talking about things, but I've got to keep, this is fungi, we're talking about fungi. So here, slime and water molds. So the mycetes. So the fungus-like protists can be divided into slime and water molds. These are generally distinct and are not closely related. So there's something that cannabis users have really got to watch out for. That's why overwatering and stuff can harbour these, the Umicota. Water molds are of many branched hyphae like fungus. However, they have cellulose cells walls and are genetically more similar to brown algae and diatoms. Let's get into it. Water molds help decompose dead plants and animals in fresh water, though some are parasitic. Okay, right, come on. I know I've got some good stuff at the end because this is one study. No, I'm not getting bacteria today. Summary. Oh. Well, that's the spores that I found out were my Penicillium, that were the Endophytes or Aspergillus that went into my cultivars. You can do that by yours as well, by putting yours, drying yours, and then putting them back into the conditions that are favourable and seeing what germinates, proliferates. That's its drying. So I cut it and seeing what came out of it, and then these dried at the end. I thought, wow, that's cool. So if these were put into the right conditions, they would germinate. Oh, there's something you don't want. Leaf curl disease. Horrible. It spreads so quickly by allelopathy as well. So if you've got any girls close, remove this one or destroy it. See this, little mold cysts sitting there again. Pretty cool. Looks like trichomes on it, but it's not. That's water. Mold cysts. Botrytis growth and development. So this is how botrytis growth spreads through the cells in the plant tissue, in the canopy, because it's an above ground in the phyllosphere. So it infects through the stomata or through an open wound. And then it can get in, start proliferating. It can also come in through the leaf or waxy cuticle. If your cell walls are not, if you've stretched the plant, for instance, if you put it under a lot of growth and stretched it, if you've used not much, if calcium hasn't been beneficial in your plant and taken up well, or possibly silica hasn't worked good in your plant, you can have mutant genetics that doesn't allow that to happen. This is another way that it can get in. So it starts on the left here and it slowly grows because it's been given the right conditions to be put into. So for botrytis, it's above, I think, 75 activity water level or 80%. So it's got to be quite humid in there. And then it will just start germinating, finding it, looking for its right spot, germinating on the surface, and then it'll bang. It'll strike through. And then you've got trouble because it gets in, it starts killing tissue, and that will send its signals down the line. You'll start to see leaves curling and discoloration, and probably before you see any of this stuff. So if one of these cells is right in the middle of your flower, this is how easy it happens. So within 3 to 4 days, it can complete this life cycle. Dampening off disease. This is Pythium's cause of dampening off disease, and you'll see it commonly in seedlings when it looks like this. You'll think, why is it just growing and it's sort of shrinking in the center? That's very common to Pythium. And I use Trichoderma hazarium to try and combat this. So that's what I used to use because it did help with it, but I've since put fungal resistant genetics in, and only unless my girls they have an immune problem or like a virus, they are susceptible to this that I've found. Oh, and they're just cool. Look at that just again. And if you've got this ugly looking wilt above, that's Fusarium, and that's another one of the Umiceta, which has gotten into your substrate and proliferated through that. So it doesn't look very good. That's what your cannabis will look like. You'll have these brown sections and be very wilty. You go, what's going on? Why isn't it to everything? That's because that section has only been attacked in that pathway through these cells. It'll slowly get through everything else. And if you cut that down, it's not really going to make a big difference because it's in the roots. You have to really treat it at the roots, dry it out, or put new endophytes in, which is one way that I've successfully gotten rid of it. And that's, yep, that's it. That's it. All right. Any questions? Hey, Lars Larson, how you going, mate? Nice to see you. What? Are you kidding, mate? You've got a piece. No way. I've seen ones now reduced to $1,800, and I so need one to do all these different types of experiments. Oh, I'm so envious, Lars. Dead set, mate. Congratulations. Good on you. Oh. Good on you. Jeez. See, this is one reason why I really want to. I try hard. I don't make any money in cannabis. I really try to do. I do respect the law. I do it legally, and I'd really like to have. I'm not good at marketing myself or selling myself and stuff like that to make money and things. And it sucks because I need that PCR machine at $2,000 for that. I need my what's the other machines I need? My ethylene meter. I need a PXRF meter. I need a few meters that are really going to take us to the next level in cannabis that they're not really doing yet. Good on you, Lars. Keep trying, keep doing your scientific stuff, mate. I can't wait to hear your results. That's what I reckon, Manix. It's excellent. Oh, it's not an RT. It's not an RT. Those are expensive. It's not a RT though. So it's still a PCR. In other words, you can put samples in and you can run it through the program and it will still electrolyze those particles. It's a thermo PXE 0.2 mil. Okay. It's not a real-time PCR, just a regular. Big deal, mate. It's a PCR. The reason why I'm going on about this is that's with all these, we can guess a lot of things, but with this PCR machine, you can go and it can test a lot of things. It can do your terpenes, it can do your cannabinoids, it can do different types of simulations that you can put it through, depends on which programs you have. If you, like me, I really want to know about what happens with processes and things like that. And to find out, this is the machine that does it on your last. Yeah, it works. It's just doesn't do colored fluorescence stuff. That's what the genotype tech is these days. So it's kind of funky, but it works. Yeah, you can do so many different things. Identify. Yeah, look. Yeah, I can test for PM resistance. There's 2 known genes. Well, that's gene. I know one of them because I've got one of them. I've got a dominant WRKY29 gene. And they're going to go, how do you know that? All right, I'll just show you a slide. I sent it to, he's all known as Xenophile. I sent it to him recently. So I'll just pull up that slide. Should be here. There it is. WRKY gene. Oh, yeah, how do I show this to you? He hasn't got any names. All right, I'll just share my screen. Present, share screen. I wonder if it'll come up because it's a Zoom thing. Are you guys seeing that? So this is the WRKY29 gene, which is the expression of the systemic acquired resistance. So if you've got systemic acquired resistance in your cultivars, it should have activated the WRKY29 gene. And these are the tests that were being put through that prove the tissue. So Lars can do these tests, mate. So you can go through and test this at the microRNA level and put it through samplings and see what's activated, what's not, and what is. Lars, mate. How much was it, please? Is it $2,000? Like I thought, you're in the States, so it's probably, what would it be to Australian? 1,200, that machine that I know of, that I can get. I got it sent it a while ago and I thought, wow, I want that. It was 100. No. Are you hooked up? Wait, in Australia. Yeah, I can get stuff. What's it called? Shipped here. That's cool. $100 it was on. Yeah. 100. Wow. Does it come with programs? Actually, because I'm holding up airtime. If you put your fungi questions in, because otherwise it's going to end. Because me and Lars are just talking by the look of it. The patents are expired. I don't follow on that bit. That might be it. Oh, because the patents have expired on the PCR machine. They've allowed people to go and produce their own. So inside the PCR, they've just got little samplers, and they've gone and used those little samplers and put them under their cheap machines. And yeah, that's where we're at now. You bloomin' beauty. So that's why that 2 gram can't come out, and now they'll be getting cheaper and cheaper. And Lars scored one for $100. Brad. Lars says he hasn't messed with the programs yet, but I'm sure it's programmed. Oh, mate. Well, you can go and you might have to, if you can't get those programs, you can go and, if you can't write them or alter them, you can hopefully get a data bank where it'll have all the programs for you and that were already written. And then you can go and just do that. Bang. I want the program to test my systemic acquired resistance. Upload it. And then get a bit of leaf tissue, grind it up, sterilize it, put it in this, do this, put it in your little sampler, press the button, and then it comes out with those graphs that we saw before. That's the level I want to be at, Lars. Good on you, mate. Keep up the good work. Yes. Anyway, I don't know what to. You've baffled me, Lars. That's pretty cool. We'll have to. I wouldn't mind getting that link somehow. Can you put links in chats? It'd be cool. It'd be interesting to see that. I'll just go into. I'm just looking at the chats. Yeah, you're a moderator. I'll add Raptor and Maine as a moderator. They weren't moderators. They're 2 cool people. Standard. Cloudscape. I'll add you. Momenix. Add a moderator. The other folk I haven't added as moderators, come in here a few times and I know that you're all right people, and then I'll add you as moderators. Momenix. Because a few, like Keith, Bond, Devil Shipwreck, I know you're new, mate. Come in here a few times and I'll add you as moderators to help with the folk that aren't so helpful. Haven't got you in for a while though. It's been very nice. Lars says, I'm going to get a centrifuge for 54 refurbished on eBay. Oh yeah, that's cool. Yep, the little things help, having little home labs. People think you can't do this sort of stuff. Lars is an example. He's just like me. He's trying to do all this stuff at home, and you can do it. You can do tissue culture. You can do all these lab techniques. You just got to create the lab conditions. So that's the only hard bit. But you've created lab or growing conditions for your cannabis, medical cannabis, so you can do the same if you want to get into these testing stuff too. Or just know people like me and Lars and hook up and send us your stuff or something like that. You can do it maybe down the track. What's Dave's 1969 say? That shows how greedy the people who had the patents are to charge that amount. Oh yeah, mate. They got to make their money off them. That's the only thing. They put all this research into the scientists and they've got to make their millions and millions and blah, blah. And that's one good thing though. Patents, at least they expire. So we do have the opportunity to get these types of stuff. So it's cool as. And Keith Bonds. Oh yeah, no worries, Keith. Thanks for that, mate. I'm not being rude. I'm just trying to monitorize the channel in the right way. I want to go do the right thing by the community as well. I don't want, because if I get people that I put as monitorizing and they monitorize people who are nice, that turns those persons off the channel, and they might have been relevant. And because I'm trying to encourage people to the channel that are intelligent cannabis users who are interested in being a cannabis connoisseur, not just in it for the money, because I'm in it for the medicinal purposes and trying to bring the best out of the genetic potential in each cultivar and trying to improve each genetics as well through different tests and experiments and stuff, and like this WRKY gene as an example. Yeah, says Lars. It was expensive hardware, just the licensing fees. Yeah, and all the programs and stuff like that, because the people got to write all the programs and things too, and that's where it comes into it, because people got to sit and do time and stuff to actually get it done, that sort of stuff. Lars says, Raptor was one who got me into tissue culture in the first place. Yeah, yep, Raptor's cool. Actually, tissue culture, I was going to do a talk on that either next week or the week after. Still putting everything together. Everyone can do it at home. It's good fun. Hey, Stony Creek, welcome. Today was talk on fungi. So if you've had fungi issues, or I also proved why you should not be dirty in your grow, not mishandle, not shake your plants around harvest. You should hang dry. You should not wet trim. I proved all those, why they're beneficial. And another thing today, which I really like to summarize on, is the microbial contamination through vaporizing and smoking of cannabis. And those charts, I'm going to probably edit out that section in the middle and make a separate video on it, because it's so important. I'm all about safety and promoting. I've done workplace health and safety in the past, a few courses on that. So I'm all about it. And it's a way that's, more educated. We can learn better and we can have more safer practices. So this is, it's a bit of a negative for the cannabis industry, what that study brought out, but it's a true one. And so that's that. So if we can work with them and with our fungus resistant genetics, try and install your WRKY gene. If you look back through my videos, I've got videos on how to install that into your genetic line. That was some pretty big things. And also about the legalization. Half of Australia will be legal soon to grow 6 plants. Yay! I'm just going to go and share that again, because I want to bring that back up again halfway through it, because I was just so stoked to see it, mate. New South Wales, Victoria and Western Australia, 6 plants soon. 6 plants. They're going to vote in Parliament, and when they vote, it'll get passed, because there's a majority in Parliament now for these types of things. All of the people who have been bought out and sold off, they're slowly getting out of it. See you later, Penny. No, I'm not going to say anything. So where's the 6 plant thing? Here we go. In those 3 states, soon might be legal to grow up to 6 cannabis plants. So that means I can start sharing my genetics. I've got, as you know, I'm a cannabis breeder. So I've got a lot of genetics I can share. And I will, with those nice folk. Not all those folk who have been causing the drama in Australia and saying bad things against me. They are not welcome. And they're not welcome to my genetics. Everybody else is. So yeah, congratulations, Australia. We're finally trying to make a bit of a headway with the New South Wales, Western Australia and Victoria. So move to those soon. So it's not just in ACT. So rad. Yes, that's what Max. I agree, mate. Lars Larson, I will never go back to mother plants. Yeah, mother plants, they reduce your cell count, your plant count too. They're hard for people with plant counts. Lars says, looks like it has 100 pre-programmed cycles. That is so cool. Hey, Lars, actually, I'll wrap the show up in a sec. Do you want to hit the link? Can you talk about me? Can you chat to me about that, please? There's the link. If you hit that soon, I'll get you up here, not live, but and then I'll end the show and then we can chat afterwards if you want. I'd like to find out more about that, because $100, I can nearly not put it into the budget, but I can maybe get it in a month's time, or a couple of months' time. I can sort of save for it. So that might be pretty cool. Stoney just got in. I'll be watching from the start. Or even just skip to the relevant bits, mate. 6 plants. It's a start. Exactly. It's a good start. Well, what it means is we're entering a grey area in Australia. So cannabis is not going to be looked upon as heroin like it used to be. It's going to be looked upon as a medicinal plant like tomatoes, or something, not tomatoes, but something that's more medicinal, so they can not get in trouble as much like they shouldn't. No, I'm not going to talk about me. What's Dave say? Have you seen anything about the bill they put in the Victorian medical patients to be able to drive? No, I haven't seen that. It's, as far as I know, it's only Tasmanians are allowed to drive with it in their system. It's, yeah. What they, I can't really say that, can I? I better not. Better not. You got to think before you say on these blooming things, because I'd like it monitorised, and I don't want any problems, and I don't want any repercussions and all that sort of stuff. Just try and spread the good, safe word. What's Lars? I can't right now. I have a pressure cooker clacking away. Oh, okay. That's no good. Oh, he says, okay, let me get my mic set up. All right. Well, it's not to come on here. It's just for you and me to chat, mate. Like, I'll end the program, and then I just want to know about your PCR. Maybe you can share a link in, and we can, I don't know. I just want to find out about it, mate, because that's taking me to the next level. I get all these studies and things from my Instagram feeds from the other people who I interact with on there, and they post all their studies online, and that they read from other, like this rad, anyway. And a person contacted me a while ago and said, I need a PCR. And I said, I know. I said, yes, tell me more. It's a cost thing. I'm poor. And yeah, so it's cool. So PCRs will take it to the next level and just allow me to do other experiments where I can test things and just give results. This is what I found to test when I did these tests. So it's pretty cool. Lars says, I turned it off. I'm trying to get the mic to work. Okay, mate, that'd be appreciated. Be nice to, actually, I don't think I've met you, Lars. I talked to you on another show before, but I don't think I've met you, so it'd be all right to have a chat, because you're a scientific as well. You're into bringing out the best in cannabis and trying to prove all these bro science into grow science. Or turn bro science into grow science, sorry. Dave says they were doing it a couple of months ago, but it's all gone quiet about medical cannabis patients driving. Yeah, I can't see it going anything good for it. It's, you know, it says in your system there, the ones in Australia they test, it lasts about 5 to 7 hours. So if you smoke in the morning, for instance, you'll be, you know, and drive at night, you might be all right. But, or even the night before you go to bed and wake up, you'll be all right. But that's about what they test for. It's the saliva they test for in your, it's a, they test for, it's a pH actually of your saliva. I don't want to not talk too much about it. So I'll put that link up there. That's for Lars to hit soon, hopefully. And then we can discuss a PCR, and then I can get one and share some more results for everybody. What else is there to talk about? Fungi. I didn't go through all the fungi section. Okay. Well, if there's any more statements or questions, I'll just quickly run through a little bit more of fungi, because I remember from my slides, I didn't go right up to the end, did I? Oh, I did. Oh, here we go. Here's one more I didn't show. Why does it do that? Can't you just bloom it open? Always use this. Oh, because it's a WB file or something. So this is how it gets in. So they were saying that Aspergillus, remember Aspergillosis is one of the biggest things from cannabis consumers, and it will get in this way. Oh my, that is so not technical. Sorry, I thought it was. Oh, so, wow. Anyway, it gets in, it goes through your branching down here, really deep into your bronchioles, and then right up deep into your bronchii, which is really deep, and that's why it's hard to remove, because once it's right up in there, it'll germinate. And you can see in here, it'll just germinate because the conditions are right, and that's what gives you some wheezing, and that's what gives you your Aspergillosis diagnosis from the doctor. That didn't help much, sorry. But yeah, that's, well, at least that's covered all my fungal bits. Okay, cool. Oh, hang on, there's been a bit more talk up here. Oh, he's got it working. Oh, Lars is in. Okay. I see that, Lars. Hang on, mate. I'll just go through a bit more chat because I didn't know if you want to be on. We can talk afterwards. Stoney says, if it is all legal, I hear tissue culture samples will be easier to send in the mail than clones. Does that sound right? Yeah. Well, don't forget, mate, you can send pollen. Yes. From pollen, you can do a dihaploidization, tissue culture. So pollen's a good way. To bring back that cultivar from pollen, you'd have to tissue culture it, or you can make an F1 and mix that pollen with another of your keepers, for instance. It worries me. I don't smoke. I take oil. Excellent. Well, for you, Dave, that's going to make sure that the extraction's been done correctly, and that maybe for your oil, you might want to put it through a pasteurization process and actually decontaminate it. So if you find your oil is put it on a spore plate. You know how I said earlier, go through this and go back through and put it on a petri dish and see if you can spore it. So you take a little bit of your sample, you would take 1 drop, you would maybe put it on a sterilized petri dish, and you'd have to sterilize your 1 ml first. So, it'd be all too hard. But anyway, see if you can do some bit of research and see if there's any types of pathogen, fungal growth in your oil, mate, because you might have to reduce that, and you can break it down by doing various techniques to do that. Yeah, says Lars. It's perfect for that. They don't need any light because they get sugar from the media. Dave makes his own stony Lars, and you can send synthetic. Yes, those synthetic seeds, they're made with calcium chloride and sodium alginate. I've put a video on how to make them, and they're really cool. They are temperature if it gets too high though, but they don't squash. You can see in the video, I've played around with them. It's like caviar. They don't squash. Good suggestion, Lars. If the other person has a flask already, put it in. Oh yeah, pollen IV. Heard of that? Yep. I'll talk about that, Stoney, with tissue culture, because I'm trying that at the moment. I keep pollen, and from my keepers, because that's how I do breed and do my crosses and stuff like that. It's a lot easier and a lot, what's it called? Organized. No, it's not random. Lars says, I'm going to try to make double haploids from pollen and anthers, but I suggest it's going to grow callus, and then it won't be able to do embryogenesis from there. That's all right. Yeah, it's sweet as. Once you grow your callus, then you change your growth medium, mate. So once you see little brown blobs growing, you start jumping up and down and go, you beauty. Wait for the cells, the parenchyma cells, to proliferate and get to about half a fingernail size or a fingernail, and then cut it. Do a subculture, and then put that subculture in another medium with more roots, more oxen, or if you want shoots, put it in a cytokinins-rich media, and that will grow those two. It's rad fun, mate. Yes, alginate. Sodium alginate is for the microbeads. Yeah, they can mail them like seeds. They are tiny. Yes, sure are. Right, I don't think it'll work though. I don't think anyone does it yet. Yeah, mate, not many people are doing the stuff that we're doing, Lars. People, PhD folk, they study one thing, so they're doctors in that one specific topic. And then they're helping other PhD candidates. So we don't have anything like that. So we do our own research and document it. So it's not many things. I've got a few other things that haven't been done. I'm playing with ploidy, with doubling chromosomes, and I've got tetraploid seeds that I've tested true. No one's talked about having that. People only talk about triploids. I can make them and breed with them, and they're 85% infertile. So there's a lot of things that cannabis folk aren't doing yet, mate, that we are. All right, chat stopped. I reckon I should stop. Oh, it's an hour and a half. Geez, microbes and things are rad. All right, don't leave, Lars, please. I'm gonna let you up, but I won't. Actually, I can let you up. I don't want to let you up and it might add to stream. Oh, so you're already there. Anyway, I'll end the stream and then I'll let you up, Lars, so don't go anywhere. I really appreciate everybody for coming in and putting in the effort to contribute and share your experience and your knowledge in chat and your nice words. So thanks very much. That everybody would not be much of a show because I hate talking to myself. I really do. I'd have ended a lot earlier. Thanks for the props, Dave. I appreciate it, mate. As soon as it gets legal, you'll be getting those. You'll be getting my genetics. You've been one of the long supporters. I appreciate that. Good on you. Yep, you'll wait. Okay, so next week it might be tissue culture. I'm not sure if we're going to talk about tissue culture or the endocannabinoid system yet. Not quite sure because I'm building up a lot of slides with the ECS on how cannabinoids and terpenes interact with our endocannabinoid system in our body and turn off pathways. So I'm not sure what's as yet. So if there's anyone who wants to make any suggestion, you're welcome to go to my what's that section under my YouTube bit? You can leave comments there, the community section. You're welcome to put suggestions there, I think. And otherwise I'll have to decide through the week. Good, well, I'll chat with Lars now about his PCR. Get excited over that. And I can speak to everybody in 7 days' time, so I appreciate it again. Thanks, everyone. Happy breeding, happy growing, and good health to you all. Bye-bye.